Assalamu alaikum everyone. Welcome back to our YouTube channel Botany. Today I will discuss the topic on nutrition, reproduction and transformation of the botany. Nutrition means obtaining energy and a source of carbon to produce the organic compound needed for cellular metabolism. Most bacteria are heterotrophs and obtain energy by breaking down organic molecules from their environment. The autotrophic bacteria either use chemicals as a source of energy or are photosynthesis. Some bacteria are autotrophs and produce their own organic compound. Are photoautotroph using light for energy, but they lack true chloroplast. Some are parasites absorbing nutrients from living organisms. Some are saprobes decomposing dead organic matter and feeding off dead organisms and waste. नॉट गोना ये हमारा थोड़ा सा लोड आ रहा है इसका हम लोड होने दें ये हो गया लोड reproduction. Prokaryotes have a single cellular chromosomes inside of the plasma membrane. Binary fission bacteria reproduce sexually and divide by the process of binary fission. In binary fission, the parent cell divides into two offspring cells that are completely identically. There is no exchange of genetic material, so the process is asexual. Transformation Transformation is the alteration of a bacterial cell's genotype by the uptake of naked foreign DNA from the surrounding environment, or transfer of cell-free DNA from one cell to another. For example, harmless Streptococcus pneumonium bacteria can be transformed to pneumonium causing cells when alive. Non-pathogenic pathogenic cell takes up a piece of DNA that happens to include the allele pathogenicity from dead, broken open pathogenic cells. Transduction transfer of genes from one cell to another by a bacteriograph, the bacteriophage. A phage infects a susceptible bacterium and injects its DNA into the host. The phage DNA proceeds to take over the host's entire metabolism, turning it into a phage factory. It then the synthesis of phage part or the components, including phage DNA, as a final step. As a final step in the phage life cycle, the various components parts in the cytoplasm are assembled into a complete phage, and the cell is broken open or releasing the newly made phage particles during the assembly process. Occasionally, rare mistakes they are made, and bit of the most bacterial DNA becomes incorporated into the new phage. Since there is no room for phage DNA in this situation, defective phage are made. However, these defective phages still have the ability to subsequently bind the insert their DNA into a host bacterial cell. But since these defective phage contain only bacterial DNA, an infection does not occur. Recombination can take place between the previous host DNA and the new host DNA, and any alleles added into the host by the defective phage can become incorporated into the host DNA, thereby changing its genotype. Conjugation. Two cells join briefly, and one cell donates some plasmid into their one. Genetic recombination in several groups through the use of Pillay minutes tube that allow the passage of the bacterial chromosomes from donor cell to the recipient cell. The transfer of genes between cells that are in physical contact with each other. Microorganisms, bacterial endoscopes, endospores. Microorganisms change and adapt to change in their environment when favored nutrients are exhausted. Some bacteria may become motile to seek out nutrients, or they may produce enzymes to exploit alternative resources. One example of an extreme survival strategy employed by certain gram-positive bacteria is the formation of endospores. तो ये था आज का हमारा टॉपिक, तो बाकी हम इसको नेक्स्ट वीडियो में इंशाल्लाह कवर करेंगे। हमारे चैनल को लाइक और सब्सक्राइब कर दीजिएगा, बेल आइकन को ज़रूर दबा दीजिएगा ताकि हर नई आने वाली वीडियो आप तक पहुँच सके। अल्लाह हाफ़िज़।